Hello, good morning friends. Today we will discuss about heme group in details. Heme group, it is a part of hemoglobin. Hemoglobin made up of the two component heme plus globin. Heme, it is a non-protein prosthetic group, while the globin, it is a protein group. What is the meaning of the prosthetic group? Prosthetic group is any group which is a non-protein in nature, which is tightly associated with the protein and which helps in the function of that protein. It is known as a prosthetic group. Function of hemoglobin is transport of oxygen. So it is actually this heme group which binds with the oxygen. So now let us discuss about the structure of heme in detail, which is our today's topic of discussions. Heme group, again it is made up of the two component, organic component and inorganic component. Organic component has a porphyrin ring, while the inorganic component is iron atoms. So heme, when we see the structure of heme in details, so we will see that it is actually porphyrin ring, it is actually the outer organic ring. This outer organic ring, which is made up of the tetrapyrrole ring, which is joined to each other by these methane bridges. So this is the outer, the porphyrin ring. When in the porphyrin ring, at the center of porphyrin ring, iron atom is present. So porphyrin ring plus iron together, it will make a heme. Okay. Now one globin chain attaches with the one heme group. Okay. One globin plus one heme, it will make the hemoglobin monomer. But we all know that hemoglobin, it is a tetrameric proteins having the 4 polypeptide chain. Therefore, the one molecule of hemoglobin has a 4 globin chain. Therefore, the 4 heme group and the one heme group has a 1 iron atoms. Therefore, the 4 heme group has a 4 iron atoms. One iron atom can bind with the 1 oxygen molecule. Therefore, the 4 iron atom can bind with the 4 oxygen molecules. Therefore, one hemoglobin can bind with the 4 oxygen molecules. The iron which is present in the hemoglobin, it is present in the ferrous state. Okay. So this is the general idea about the heme group. Organic protoporphyrin ring, and at the center of that iron atom is present. So now let us see this structure in detail. So just we had discussed that the structure of heme contains a ring of a tetrapyrrole. The tetrapyrrole is formed by joining the 4 pyrrole rings by methane bridges, and that will make the structure of the porphyrin rings. So now let's see what is one pyrrole first. So one pyrrole has a 5 atoms, 4 carbon and 1 nitrogens, and all these are arranged in a shape of a pentagons. So that will make a 1 pyrrole rings. Heme has a 4 pyrrole rings. So these are the 4 pyrrole rings, and these are all the where we have not written anything. Those are the carbon atoms and 1 nitrogen in each ring. Now let us draw this structure one by one. So just we had seen that all these 4 rings are joined to each other by the methane bridges. So first we will form a bond here, and then later we will see the position of double bond. So first we will make all the bonds for the methane bridges. Okay. So this is how all the 4 carbon and all 4 pyrrole rings are joined to each other. Now we will see the position of double bond. So how to remember this? For the upper and the down side, it will be the symmetrical, means or both the double bond will be at the right side. Okay. So one double bond will be here, one double bond will be here, and for side to side, both the double bond will be at the down side, means here. Okay. So the position of the double bond will be the symmetrical for these bridges, right side for the up and down and the down side for the here. Now we will see the position of the double bond for the ring. So first ring has a 2 double bond here. Second, third and fourth will have the 1 double bond each at the back side of the ring. For this ring, as the carbon valency is already satisfied because of the presence of the two double bond here, so this ring will have only the one double bond, but this means second and the fourth ring will have the two double bond. So here will be the second double bond, and for this ring here will be the second double bond. So that is how the position of the double bond, two double bond in each ring, three ring, one double bond in the third ring, and this is the position for the methane bridges. So 4 pyrrole rings connected to each other by the methane bridges. Heme has a 4-methyl, 2-vinyl and the 2-propionate group attached as the substituent of pyrrole rings. Now let us see the position of that. So 4-methyl. So here will be the first methyl group, second methyl, third methyl group, and the fourth methyl group. 4-methyl group, M is for methyl, 2-vinyl, 1 and 2, 2-vinyl group and 2-propionate. So at the bottom 2-propionate group. Okay, so this will make the porphyrin ring. Okay, but we know the heme, it's made up of the porphyrin ring which has the iron atom at the center. So here the iron is present, which is present in the +2 oxidation state, means it is present in the ferrous state. Okay, there are two forms, ferrous and the ferric. Ferric has a +3 oxidation state, ferrous has a +2 oxidation state. So heme group in the hemoglobin where the oxygen is not bound, so in that case iron is always present in the +2 oxidation state, and an oxygen molecule can bind to the iron only in the +2 oxidation state of the iron. Now we will discuss about the bonds formed by iron atom in hemoglobin. Iron can form the total 6 bonds. 4 bonds it will form with these nitrogen atoms, and which will be in the plane of these porphyrin rings. 5th and the 6th bond will be the above and below the plane of the rings, that we will discuss in a after some times. So first we will see the position of the 4 bond which it will form with the nitrogen atom of the heme group. See, these nitrogens and these nitrogens, both of them has the double bond here. So here the lone pair of the electron will be present. Okay, and this lone pair of the electrons will be form the coordinate bond with this iron atom. So here one coordinate bond is present, second coordinate bond, and that we will demonstrate here with the dotted lines, because it is a coordinate bonds which will be formed between this lone pair of electron between the nitrogen and the iron atoms. These two nitrogen atoms, it has a no double bond here, therefore its valency will be satisfied with the normal covalent bonds, and that we will demonstrate here with the solid lines. So that's how iron will form the 4 bonds with the nitrogen atom of the heme group. Okay, so this is the complete structure of the heme group. Okay, 4 pyrrole rings which is attached to each other by the methane bridges. Okay, these are the position of the double bond which we had discussed. Substituent the methyl, vinyl and the propionate. Okay, now the 4 bonds which is formed by the iron atoms with the nitrogen of the heme groups. So this is the structure of the heme group. Now just we had discussed that the iron can form the 2 more bonds. Okay, so there is a space for the 2 more bond to be formed by the iron atoms. So the 5th bond, with the 5th bond, this heme group will attach with the globin chain. So we know that, suppose if we consider as a flower, so this is a heme group and heme group will attach with the globin chain. So the 5th bond, it will form by the iron atom and the globin chain, and that is how the heme and globin chain will attach to each other. Okay, heme and the globin chain will attach to each other. So that will be by the fifth bond which is formed between the globin chain and the iron atoms. Let us discuss how. So now instead of drawing the whole structures, let us understand this as a hemoglobin molecules which has a 4 globin chains, 2 alpha globin chain, 2 beta globin chains. At the center heme group is present, and each heme group has a iron atoms which is bonded to the 4 nitrogen atoms. Now let us take the one box here. Okay, so suppose this is a one porphyrin rings where the 4 bonds are already formed with the nitrogen atoms. Now we will discuss the fifth bond, which we had seen that with the fifth bond this heme group will attach with the globin chain, and the bond will be formed between the proximal histidine of the globin chain which has a nitrogen atoms and which has a lone pair of electrons. So that fifth bond will be formed between the lone pair of electron on the nitrogen atoms and with the iron atoms. So that is how the heme group will attach with the globin chain. Okay, so total the fifth bond formed here. Sixth bond will be formed between the iron and the oxygen. Okay, diatomic oxygens will bind with the iron atoms. So that is how the total the 6 bond will be formed. Oxygen, just we had discussed, can bind only with the iron when it is present in the +2 oxidation state, and after binding with the iron the molecule formed will be the oxyhemoglobin. Hemoglobin, after binding with the oxygens, it will form the oxyhemoglobins, and after binding the oxidation state of the iron will temporarily shift from the +2 to +3, that is from the ferrous ion to ferric ions. And as we know that oxygen is more electronegative, so it will shift the electron pair towards itself and form the negative charge. So oxygen will form the superoxide ions. So to stabilize that negative charge on the superoxide oxygen, because as it is electronegative, so it will pull the electron pair towards itself. So here the one more bond means to stabilize it, distal histidine of the globin chain, proximal histidine form the bond with the iron atoms. With the distal histidine, it will stabilize the negative charge on the superoxide ions. So here also the one more bond formations, but for the exams when it will be asked, so these 6 bonds are more important. 4 with the nitrogens, 1 that will be at the plane of this porphyrin rings, 1 below and the 1 above the plane of this ring, and the this is to stabilize the negative charge which is formed on the superoxide ions. So that will be by the distal histidine of the globin chain. Okay, so this is all about the structure of the heme group in hemoglobin.